It is 1250 beds, sir.
Oh, quite a big hospital.
It's big, sir. Big, sir.
Mm.
Surrounded by only villages, sir.
Yeah, I know you are very committed to the rural upliftment as far as healthcare is concerned. Some people claim that, nothing was done till BJP came to power. That's all unfair. See, we started all the rural health mission, urban health mission, vaccination, and so many things were done, but it's such a poor country, things can happen only slowly.
Every government has done something, sir. Then
Yeah.
only it has come to certain stage.
That no? Okay. Sir, with the permission of Honorable Vice Chancellor and Chief Guest Professor T Raiju, we are going to start now. I over the mic to the coordinator of the Research Club, Department of Physiology, Dr. Jyoti Khodnapur, Associate Professor. Jyoti.
Good morning, you all. Respected Chief Guest Dr. T Raiju, Dr. A S Paintal, Distinguished Scientist Chair of ICMR, NIMHANS, Bengaluru, Honorable Vice Chancellor Dr. M.S. Biradar Sir, BLD, Dean to be University, Dr. Arvind Patil Sir, Principal, Dean Faculty of Medicine, BLD, Dean to be University, Dr. Manjuna Taithala, IQAC Coordinator, my teachers and my dear friends. I, Dr. Jyoti Khodnapur, on behalf of Department of Physiology and Research Club, welcome you all for the 38th Research Club meet on the occasion of Professor A S Paintal 95th birth anniversary, who is the first Indian medical scientist to become Fellow of Royal Society. Now I request Dr. Sumangala Patil, convener, head of the department, to welcome the gathering and to introduce our chief guest, Dr. T Raiju.
A very good morning to all and hearty welcome to this event. So I welcome our chief guest, Dr. Trichur R. Raju sir, and Honorable Vice Chancellor, Dr. M.S. Biradar sir, in Faculty of Medicine, Dr. Arvind Patil sir, and all our faculty members of the department and BLD University Shri B.M. Patil Medical College and the dear students. So it's my privilege to introduce Dr. Trichur R. Raju, who is a AS Paintal Distinguished Research Chair of Indian Council of Medical Research. Sir is a senior professor in Neurophysiology, National Institute of Mental Health and Neurosciences, Bangalore. And Sir completed his MSc in Medical Physiology from JIPMER, University of Madras in 1974 and PhD in Experimental Pathology, Applied Physiology from John Curtin School of Medical Research, Australian National University. Postdoctoral training at Harvard Medical School in 1982. Sir's research work is focused mainly on the developmental neuroscience to understand critical roles of trophic factors, glia, neurotransmitters, and intrinsic activity in neuronal survival, growth, and formation of synaptic connections in the developing retina. They are phenomenal. Sir is also one of the pioneers in describing the cytoskeleton and cell motility in the world, and his contribution towards understanding neuronal plasticity in the nervous system and remodeling of brain circuits in treating stroke and depression are noteworthy. Sir has also done a great contribution on central nervous system regeneration and resident stem cell activation in spinal cord that will help greatly in the treatment of paralytic and depression patients. T Raiju sir has also worked on preclinical models on rodents for the neurological diseases, exploring new therapeutic modalities for the treatment in neurodegenerative diseases like amyotrophic lateral sclerosis. Sir is also an expert member of Indian Council of Medical Research, National Center for Biological Sciences, NCBS, Institute for Stem Cell Research, and International Institute for Training and Research in Reproductive Health. He is also an expert advisor to various government and non-government organizations. And the use of complementary medicine in treating mental health and neurological disorders, exploring the power of yoga, Ayurveda, and naturopathy to treat Parkinson's disease, ALS, stroke, and multiple sclerosis. Sir has more than 200 publications in the reputed journals and book chapters to his credit, with H index of 45. And Sir has also received various awards and honors, highest Indian honors in the neuroscience and physiology. So here I present you one of the greatest neuroscientists of India. Welcome you, sir.
Thank you so much. I'm deeply humbled by your introduction.
Thank you. It is nothing, sir. Very brave.
Thank you so much, ma'am. Now I request Dr. Srilakshmi Bagli, Associate Professor, Department of Physiology, to enlighten us about discoveries of Dr. A S Paintal. A very good morning, everybody. I consider this day as an auspicious day for all the physiologists of the country and across the globe, the 95th birth anniversary of Professor A S Paintal. He is a legendary physiologist and a towering figure who has contributed immensely in cardiovascular physiology. And he is a world-renowned scientist who has made breakthrough discoveries in the field of cardiovascular and pulmonary reflex. Coming to his journey as an early researcher, he did his MBBS from King George Medical College, Lucknow, and during UG years, it was a period of honors, achievements, and awards, ending off with the most coveted Hewitt Gold Medal for obtaining highest marks in the final MBBS. During the undergraduate years, most of the time he was found under the bright lamps of the Lucknow Presidency grounds, so that he finds a quiet place where he can concentrate on his studies. So with this enviable performance, most of the people were of the opinion that he would continue his career in clinical medicine. But in quest for scientific research, he gave up the lucrative future in clinical medicine and pursued his career went to the Department of Physiology and did his MD in Physiology. So this was the opinion which is recorded by S.N. Mathur, who was the professor of physiology. He has stated that I've kept the sentences verbatim so that it doesn't lose the essence. In my lifelong experience as a teacher, I have not come across another young man who, through sheer love and liking for research, has deliberately given up his clinical training for the sake of a research job. During his MD period, he chose the subject of psychophysiology and undertook the study of measuring the electrical resistance of the skin in normal and psychotic patients. For this, he developed an instrument all by his own by using the junk that was available in the free market, and was able to give some very good results, which came to be known as Paintal Index. And this Paintal Index was used for a long time for the diagnosis of psychotics. Still, some other advanced techniques came into force. Then once he completed his PhD, he immediately applied for a Rockefeller Fellowship and went to Edinburgh University and pursued his PhD career under the illustrious guidance of illustrious physiologist Professor David Whitridge at the University of Edinburgh, and made some breakthrough researches. And because of his tremendous contribution, Nobel laureate Cornel Hammonds and Eric Neal coined the term pre-Paintal era versus post-Paintal era. So these are his noteworthy scientific contributions, the most important being the single fiber technique. So this single fiber technique made him to undertake several other researches and led to the discovery of type B atrial receptors, gastric stretch receptors, juxtapulmonary capillary J receptors, ventricular pressure receptors, mucosal mechanoreceptors of the intestines, and pressure pain receptors of the muscles. So these are some of the pictures of his during his Edinburgh days. And the middle one is a picture where he
himself is making the instrument for the electrophysiological studies, and the last one is he's enjoying the looking at the recording that he has performed on the previous day. So this single fiber technique is an extremely tedious job of separating the single fibers or nerve fibers from multi-fiber bundle. And in those days, steam box was used for the single fiber technique. So in this technique, the experimental animal, that is the cat, was placed in the box and the steam was continuously generated so that whatever nerve fibers are separated, they don't get dried up. But the most annoying disadvantage was that the condensate of the steam on the microscope made it difficult to see the nerve fibers while dissecting. So Paintal came up with the innovative idea and he went and informed his supervisor, Dr. David Bittridge, that he wanted to use liquid paraffin, immerse the nerve fibers in the liquid paraffin and then dissect it and record the impulse. So everybody warned him that if it is a failure, then he has to book the ticket back to Mumbai. But A.S. Paintal was confident that he'll get through, and he was able to successfully dissect the nerve fibers under liquid paraffin and record the impulses. So this technique was highly appreciated, and from then onwards, it was being used. It is, and it's still in use in most of the universities in the UK and US. And the second important, once he finished his PhD, he took up the work of identifying the location of the pulmonary vascular receptors, which were supposedly discovered by his supervisor, Dr. David. So once he started working on the experimental animal, that is the cats, he was shocked and surprised to see that the pulmonary vascular receptors didn't exist. And instead, he found the receptors subendothelially on the posterior aspect of the right atrium and the left atrium, and also near the veins, and not the pulmonary vascular receptors. Now he had to inform this to his supervisor, and he performed a similar experiment several times, and he got similar results and informed it to his supervisor. And the supervisor graciously accepted this fact, and he was very happy that his student has made a correction. And since the type A atrial receptors were already there, and these set of receptors were different from the type A receptors, he named them as type B atrial receptors, which were slowly adapting stretch receptors, which were sensitive to the atrial filling, unlike the type A atrial receptors. And his mentor himself presented this and in a meeting in a physiological society, and this is the publication. And interestingly, this publication is having the name only of A.S. Paintal, and it was during this time he learned, his mentor taught him the games, rules of the games and rules of the publication, which he followed it throughout his life. This is a very interesting article, and it is available free online. The second breakthrough discovery in the field of cardiovascular reflexes and the pulmonary reflexes is the identification of J receptors. So this J receptor, when he came back to India, when he joined the VP Patel Chest Institute of New Delhi, he undertook this research program where he started identifying the location of the J receptors. So this is considered as a very important research in independent India. So initially, when he found the J receptors, he named them as deflation receptors. And as he was working in this laboratory, one late night, he found that all nerve fiber showed impulses after he injected the chemical phenyl diguanidine in the right atrium. Once he got that, he proposed that there are receptors, free nerve endings, which he named as deflation receptors. Later, he published a paper and proposed that these receptors are present distal to the respiratory bronchioles. He continued the work on J receptors for the next 12 to 14 years, and at the end of 14 years, he proposed that the J receptors, the deflation receptors, are actually the J receptors which are present in the interstitium. Which are present in the interstitium close to the alveoli and the pulmonary capillaries, and named them as J receptors, juxtapulmonary capillary receptors. And he also showed that whenever the J receptors are stimulated, there is reflex bradycardia, hypotension, and tachypnea.
and demonstrated that these J receptors are stimulated by pulmonary congestion, particularly during moderate exercise at high altitudes. Usually, otherwise, in normal healthy individuals, these J receptors, they are not active. So, a breakthrough point in the discovery of J receptors was he attended a symposium in SIPA symposium in London in 1969 to attend the centenary celebration of Herring Brewer reflex. There he met a physician by name Abraham Gus, who introduced A S Paintal to his colleague Lynn Reed. And Abraham Gus persuaded Lynn Reed to demonstrate, to take an electromicroscopic study to demonstrate if the receptors proposed by A S Paintal are present in the interstitium of the lung. And Lynn Reed actually demonstrated the presence of these J receptors in the interstitium in the rats, and later it was demonstrated in mice and man. And in turn, A.S. Paintal volunteered to be the subject of Abraham Gus for his next research, which was on finding the respiratory manifestations of intravenous injection of capsaicin. It is a principal pungent material which is present in capsicum. So this is the picture showing him getting ready for the shot of capsaicin. And this is a classical record of the classical science, where the scientist working in his laboratory at the dead of the night, immune to the passing of time and the fact that he is semi-nude. And this is in the laboratory of Delhi, getting the vagal nerves ready to record from. Though he was very much interested in research, he was also a sting teacher. So his students described him as an enigma, a brilliant, fascinating, witty, explosive character with more than a trace of resemblance to the action potential. So this was the extent to which he enjoyed the teaching. And most of the teaching he did was when he was a professor in All India Institute of Medicine. And he was an avid collector of limericks. He enjoyed bird watching, he enjoyed rowing, he enjoyed swimming. And though most of the times he was with his instruments, repairing them, getting ready, preparing them for the next experiment. But however, whenever he found free time, he went for rowing and enjoyed bird watching. This is one of the pictures. And he was also honored by a number of fellowships and awards. He received the prestigious Dr. B.C. Roy Oration by MCA in 1974. And he was awarded, he also became or elected as Fellow of Royal Society of Edinburgh in 1966, followed by National Academy of Medical Sciences 1966, Fellowship of Indian National Science Academy 1972. And he was the first Indian medical scientist to be elected as a Fellow of Royal Society UK in 1981. He was also honored with Padma Vibhushan, Fellow of American Society of Physiology, and he was the president of Indian Science Congress and Indian National Science Academy. The list continues, but I have highlighted the most important ones. And recently, my mentor, Professor Kusal Das sir, had requested Professor Nanduri R. Prabhakar, who was the fourth recipient of A.S. Paintal Memorial Oration, to summarize briefly what he thinks the most important contribution of A.S. Paintal in the field of cardiovascular physiology and pulmonary research. And this was the message that was sent by him. He has summarized briefly and very beautifully in just 2 sentences. Development of single fiber technique along with measurement of conduction velocities led to the identification of sensory nerve fibers. That too, he could differentiate between the modulated and non-modulated nerve fibers. So dissecting single nerve fibers is a very tedious job, particularly the non-modulated fibers are very minute. So what he did was he found that the modulated fibers could be blocked at 7 degrees centigrade. So he maintained the temperature of the nerve fibers at 7 degrees, and then he recorded the myelated action potentials from non-myelated nerve fibers. It could differentiate the conduction velocities. It could differentiate the difference in the spike potential. And the second most important contribution was identification of J receptors that led to the proposal of non-modulated C fibers subserve nociceptive function. So the research work of A.S. Paintal has shown a path for the younger generations to take up research in cardiovascular and pulmonary reflexes. And this is a beautiful picture which was captured during his visit in 2003 to Al-Amin Medical College. And this picture is
It is a, I feel it is a very important, interesting picture. Why I'm sharing it here is it is showing three important people whom I am inspired by. One is Professor A S Paintal, legendary physiologist. His student B.N. Cole, sir. Professor B.N. Cole, he is again has been mentored by Professor A S Paintal. And my mentor, Professor Kusal K. Das, who has been mentored by Professor B.N. Cole. So it is a legacy where my mentor, Professor Kusal K. Das, has transferred it to us. And in an era where the experiments on experimental animals is dwindling, Professor Das has motivated the younger generations to take up research involving the experimental animals and kept alive that legacy. And I end my talk by conveying that the entire fraternity in biomedicine shall remember the seminal contributions of Professor A S Paintal with gratitude and reverence. Thank you.
Thank you, madam, for enlightening us. Now I request Dr. T Raiju sir to enlighten us about in the world of science.
Okay. Let me do the screen sharing first. First of all, let me thank the Honorable Vice Chancellor, respected Professor Das.
The slideshow. You go to the full screen. Can you able to go to full screen?
Yes, I can.
Yes, sir. Thank you very much.
Not to worry. So, Professor Sumangala and other esteemed members of the BLD community, I sincerely thank you for giving me this privilege to address you on this auspicious day when we are remembering Professor Paintal. My previous speaker has done a wonderful job summarizing all the academic achievements of Professor Paintal. And I'm also very impressed that he proved his teacher wrong. I must say very bright students always prove the teacher wrong. And for that, they really work hard though, as Paintal showed. So I'm not going to repeat what was said before and all this, he was a very brilliant student, and pre-Paintal versus post-Paintal era, and all the awards, this commitment to working on J receptor. It's really amazing. And one thing about Dr. Paintal is he always believed that there should be a technological innovation in the country. We should not keep on importing equipment. And he was so worried, concerned that there is a lack of technological innovation in the country. And when he addressed a convocation at the IIT Kanpur, he said, we have given up self-reliance as a driving force. You thought of Atmanirbhar Bharat even those days. Isn't it amazing?
Absolutely.
And that he was very concerned about, the import. And at that time in the 1962, the war was there, Indo-China war. He had such a soft corner for soldiers who were suffering at the high altitude. And he described this high altitude pulmonary edema and how the J receptors could play a role in producing dry cough and muscle weakness and other cardiac issues associated with living on the high altitude. He really wanted the government to create an institute for mountain research or high altitude studies. He strived so hard, but that dream could not be fulfilled. And another interesting about Paintal is that he will call a spade, and very few scientists could do it. And he formed the Society for Scientific Values. And he would say that, if he believes that somebody's work is not dependable then he'll go and say that, I'm sorry, this is all a load of bull. He will say that.
Yes.
So that is his, that is his conviction, commitment to values in science. And he was also concerned about the way the ethics committees were functioning in India at that time, right and left giving approval for clinical research. And also, this is something, there was no accountability of the members of ethical committee. That was, he felt very, very bad. And the clinical trials, he thought that we should look at them very, very carefully. What is the benefit, what is the serious adversary event? And these are some things which we need to appreciate. And when he became the Director General of Indian Council of Medical Research, I would say it was a tumultuous period. At that time, the HIV was raging through India and it's coming. He thought that because it's imported from visitors from abroad and NRI. And he made some controversial statements. So when he made these controversial statements, people were just making fun of him. Then what he said was, if you say a DG is very good, nobody is going to bother. When you say if DG is full, they'll start reading it. Along with that, they will also come to know about the importance of HIV. That was his attitude. And when he said he is the biggest ass on earth, everybody is going to read. It was so funny. And the other thing is, he always felt that research should not chase just publications and citation index. Research should have a social value. Research should have an application, and it must address the alleviation of suffering humanity. It should focus on finding some new knowledge in a human disease. That is very important. This takes me to my work, which is on the translational research, the trials and tribulations. And the disease which we addressed was what is translational research? Translational research involves transformation of basic research into development of new drugs or new therapeutic modalities like stem cell therapy, or finding out new biomarkers which would objectively diagnose the disease and explain its pathogenesis. Which disease we focused on? We focused on a disease called amyotrophic lateral sclerosis. We just wanted to follow Paintal. We thought that, we should really address... Hello, what's happening?
No.
Is everything okay? So we wanted to do some translation research in amyotrophic lateral sclerosis, hoping that our research would make a difference to these people. And this is a crippling, devastating, and debilitating neurodegenerative disorder. And this is a disease with no cure. And this is a relentlessly progressive disease. Patients die within 2 to 4 years. This is traumatic and shattering experience for caregivers. The patients come with muscle weakness, fasciculation, dysphagia, dysarthria, and respiratory muscle paralysis. The patients die. So we have been pursuing this research for the past 2.5 decades, hoping against hope that we will find something which will make a difference. So I want to show you a video here. This is a patient by name Alex. The kind of thing he went through as an ALS patient is beautifully captured in this video and give you a feel for the magnanimity of the disease. Can you hear the sound?
Give me a thumbs up with your other hand. Can you lift it up? There you go.
His muscles are getting weak, but he still doesn't give up.
Listen to that. Cool. You guys love football? Yes. Yes. Ready?
Look at also the stress on the caregiver. Muscle weakness, wasting. In general, it affects people with 50 years or older, but there are cases in which younger people are also involved. he takes an ice bucket challenge. This was promoted to get funding for ALS research. You take a bath in bucket full of water with ice. And people on seeing this will start donating funds to ALS research. Alex does it. It's quite amazing. And when everything becomes almost impossible, he doesn't give up, does he? He has faith in God. Then he dies. So all these things happen within a period of 2 years. So after seeing the sufferings of patients with ALS, I thought 2.5 decades back, we should do some research in this to understand the pathogenesis of this crippling disease. What was our hypothesis and approach? We strongly believe that CSF from ALS patients contains toxic factors which could be lethal to motor neurons. So interestingly, in this disease, only the motor neurons are affected. Sensory neurons are spared. So what was the experimental sample? We took CSF from ALS patient after obtaining the ethics committee approval and the informed consent, because we thought that CSF has toxic factors against the motor neurons. The control samples were CSF from other neurological diseases. The patients who do not have ALS or other neurodegenerative diseases and neuroinflammatory diseases. This is a neurological control. And normal control or close to the normal control was CSF from orthopedic patients undergoing spinal anesthesia. So we used cellular models as well as animal models to understand the pathophysiology of this disease. For cellular model, we had primary cultures from embryonic rat spinal cord, and we used a motor neuron cell line, NSC34, which is a fusion between motor neurons in culture plus neuroblastoma. So it is an immortalized cell line. Then human embryonic stem cells we obtained, and we derived motor neurons out of human embryonic stem cells because it's a human disease. So we have to work on human cells too. And for animal models, we use newborn rats and adult rats. Newborn rats were intrathecally injected with ALS CSF or control CSF, and the adult rats were intracerebroventricularly injected with ALS CSF. When you look at the primary cultures of neurons, which are motor neurons, which are exposed to non-ALS CSF on one hand and ALS CSF on the other, you can see these motor neurons in the non-ALS CSF, they are so healthy, face, bright cell body, and lot of processes are coming out. But if these neurons are exposed to, sorry, something, am I audible?
Yes, yes.
Okay. If these neurons are exposed to ALS CSF, you can see the beating of neurites. that's an indication that the neurons are degenerating, condensation of the cell body, and evacuation of the cell. And this is the motor neuron in culture. I can hear some background noise. Sorry, this is a motor neuron in culture. It looks so beautiful, that cell soma and a long process. And these neurons are exposed to ALS CSF and labeled with an antibody called SMI31. It labels phosphorylated neurofilament. And you can see the beading in the processes. And the other important thing is that this antibody in a healthy neuron should not label the cell body. And here you can see it is labeled. That means although it is apparently healthy looking, this neuron is undergoing degeneration when exposed to ALS CSF. So this is NSC34 cells, immortal cell line, and we used tunnel staining as well as caspase-3 labeling to understand whether these neurons undergo apoptosis following exposure to ALS CSF. Normal control with ALS CSF, more tunnel labeling indicating more DNA fragmentation. And caspase-3 also is enhanced in the presence of ALS CSF, which clearly shows here in the histogram. So tunnel positive cells are more, and caspase-3 labeling also is more, indicating that these cells undergo apoptosis when exposed to ALS CSF. Then we come to motor neurons, which are derived from human embryonic stem cells. And this is a tough procedure. Once we get the embryonic stem cells, and we didn't produce these embryonic stem cells, we got it from another source after obtaining all the approvals, but we derived motor neurons out of this human embryonic stem cells using TGF, bone morphogenic protein inhibitors, retinoic acid, plus SSH agonist, and different neurotrophic factors. And we could get these process-bearing motor neurons from human embryonic stem cells after 27 days. And we labeled them for CHAT, cholinesterase transferase, which is expressed only by motor neurons, and beta-3 tubulin, and this is the merge image. And this is a confocal photomicrograph after 21 days of differentiation of human embryonic stem cells into motor neurons. And the proof of pudding has to come from eating. So in this case, we have to demonstrate the electrical activity in these derived motor neurons, which we managed to do by using a multi-electrode array. And when you look at the activity, the action potentials which come from the cultures where there is a presence of motor neurons from human embryonic stem cells, and analyze the activity, most of the activity was less than 5 Hertz when you look at the average frequency. If the neurons fire less than 5 Hertz frequency, that means they are projection neurons. That means in our case, they are motor neurons. Few of them could be undifferentiated motor neurons. It's a possibility is there, but we were very happy that more than 80% were firing less than 5 Hertz frequency. So we could derive motor neurons from human embryonic stem cells, and we published this study. This was the first study in this field. And when these cells are exposed, when the motor neurons derived from human embryonic stem cells were exposed to ALS CSF, there was a decrease in the cell viability, which you can see, and there was an increase in LDH assay, indicating that the cells are undergoing degeneration. So there was beading of neurites we could see as well as vacuolation in the cell soma. To sum up, we could get motor neurons from human embryonic stem cells, and we could show that these neurons undergo degeneration following exposure to ALS-CSF. So if this is only in culture we could see it, or is the finding extended to in vivo? So we used newborn rat pups, intrathecally injected the ALS-CSF, took spinal cord sections, and if you look at the ventral horn here, the non-ALS-CSF and the ALS-CSF. We label again with SMI31, it's an antibody against phosphorylated neurofilament epitope. If the neuron is healthy, the soma should not be labeled. You can see these empty spaces here. And nice neuritic processes or axonal processes, which you could see. But if the animal is exposed to ALS-CSF, the motor neurons start labeling in the cell body, and also there is a severe disruption of neuronal processes. So we could show it in the in vivo as well. And it's not only the neurons, motor neurons, which are affected. So there was also a reactive gliosis. How did we show it? We did a double labeling. In this case, red for GFAP, which is a specific marker for astrocyte. And here, we use green labeling for glutamate transporter. And in the non-ALS-CSF exposure, you could see these GFAP activities restricted to white matter and bright labeling of GLT. But when it is exposed to ALS-CSF, there is a change, significant change. Intense labeling of the white matter, plus you can see gray matter also gets labeled. So the reactive astrogliosis occurs in the gray matter as well. And there is also a down regulation of GLT1 compared to non-ALS-CSF. So reactive gliosis and GLT1 down regulation. The glutamate transporter is an important molecule which is involved in the reuptake of glutamate after its action is over on the motor neurons. But if it is not, if there is a depression of, there is a suppression of the uptake, the glutamate will hang around in the synaptic cleft for a longer time and keep on driving the motor neuron. So that will result in excitotoxicity. So our results clearly show that ALS-CSF downregulates the expression of GLT1, which is a bad news for motor neurons. So what happens to the astrocytes? Many things you can't look in detail in vivo. Then again, you go back to in vitro culture. It's a primary cultures. I miss live audience, I must say. So here, when we grew the astrocytes in culture, in the non-ALS-CSF, they look epithelioid in nature, and this we call protoplasmic astrocytes. These are neurotrophic astrocytes. If you grow neurons on the top of them, the neurons will grow so beautifully. If you take conditioned media from this culture and put it into the cultures of neurons, it will grow beautifully. So this is neurotrophic astrocyte. But when you expose them to ALS-CSF, there is a dramatic transformation in morphology. They become process-bearing fibrous astrocyte. Neurotrophic becomes neurotoxic, which is brought about by ALS-CSF. And another important glia is microglia. The microglia in resting state, unlike the astrocytes, it is in the fibrous shape or ramified. These are called M2 microglia. But once you expose them to ALS-CSF, they become activated and they become amoeboid in shape. So if you look at the percentage of amoeboid cells, this is normal control, non-ALS, ALS-CSF. there is a lot of these amoeboid shaped cells which are formed because of exposure to ALS-CSF. I'm going to show you a movie here. This is microglia grown in normal condition media. You can see that all is ramified microglia representing M1, M2 type. But on exposure to ALS-CSF, look at, they become huge amoeboid. See this cell which is migrating from here to there. So to summarize, in vitro and in vivo models reveal CSF from ALS patients induce selective vulnerability of ventral horn motor neurons, severe organelle dysfunction. I'm not showing you data on this because of the time being short. Mitochondria are affected, lysosomes are affected, endoplasmic reticulum and Golgi apparatus also are impaired following exposure to ALS-CSF. There is a down regulation of ion channels like voltage-gated sodium and voltage-gated potassium channels, aberrant neurofilament phosphorylation, abnormal cortical motor neuron firing, apoptotic and necrotic death of neurons occur. So there are neuronal changes. Come to glial changes, morphological transformation of microglia and astrocytes, I showed you evidence. Decreased glutamate transporter, GLT1, I showed evidence. Enhanced oxidative stress parameters, which I didn't show because of lack of time. ROS, reactive oxygen species, nitric oxide, iNOS, they are expressed more following exposure to ALS-CSF, and increased glutamate release. So increased glutamate release and decreased glutamate transporter is double whammy for motor neurons. More glutamate is released, less uptake, and a lot of glutamate is driving the poor old motor neurons. And elevated pro-inflammatory molecules like PGE2, COX2, IL-6, TNF-alpha, decreased anti-inflammatory molecules like IL-10, IL-4, and reduced trophic factors. VEGF, GDNF, and BDNF also occur. So you have an environment where there is a lot of pro-inflammatory molecules, increased release of nitric oxide, increased secretion of glutamate, reactive oxygen species, and decreased glutamate transport, and reduced trophic factors, which are critical for the survival of motor neurons. So the ecosystem or the microenvironment is too bad for motor neurons to survive. And it is not only the glia and motor neurons, the muscles also get affected. Type 2 fibers, the fast muscle fibers are significantly affected. The neuromuscular junction becomes disrupted, diffused, fragmented. So neuromuscular transmission is affected very badly. And because of this decreased motor performance, we tested with rotor rod and grip strength, muscle weakness, and atrophy of muscle, all these things take place. And I want to show you the video. For this, I need to do a circus because this is an old video. My Mac won't take it that easily. So hang on for a moment. This is a rotor rod. We put rats on a rotating drum. And the third one from the left is injected with ALS-CSF. All the others are different controls. When she increases the speed of the rotating drum, And because of the weakness and lack of muscles, the poor guy falls down. So we demonstrated not only structurally, biochemically, but even with motor behavior. I want you to see it again. Focus on the third one from left or second one from the right. It is unable to balance because of muscle weakness. Yeah. Then I'll go to my slides. So what is it in the ALS CSF which produces impairment to the motor neurons, changes the phenotype of glia, and affects the muscle? So we went on to do a lot of serious biochemical experiments. We used proteomics, and we found in the ALS CSF there was a 10-fold increase in a protein called chitotriosidase. And there was also a slight increase in osteopontin, chitinase 3, like protein 1 and protein 2. But significantly, see, in science, you can't chase everything. You take bets in science. Sometimes you put your neck on the chopping board and it gets cut too. You have to be careful. Otherwise, the science is boring. If you start chasing everything, it becomes a stamp collecting exercise. So we thought that we would focus against the wishes of my lab members. I thought that no, we are going to focus only on that. This is chitotriosidase 1. So we did some ELISA on lots of samples, ALS CSF samples, and then non-ALS CSF samples and normal samples. These are from orthopedic patients. We managed to show this, the cohort size is 158. Right now we have a cohort size of over 200, which we published very, very recently in Journal of Neuroinflammation. So there was 20 times more expression of chitotriosidase in the CSF of ALS patients. The chitotriosidase is an enzyme, so we want to look at the enzyme activity. Even the enzyme activity was 16 to 18 times more compared to non-ALS CSF or normal CSF. So we have a putative biomarker here. And we want to claim it as a diagnostic marker for ALS because CHIT1 level can distinguish ALS patients with a sensitivity of 87% and a specificity of 83.3%. That's a protein level. Enzyme activity of chitotriosidase has a sensitivity of 80.4% and specificity of 80% in identifying ALS patients. Combining both the protein and the enzyme levels gave a positive predictive value of 97.78% and a negative predictive value of 100%. I would say it's reasonably good in anybody's language. I hope I'm true. So the other benefit with this marker was, which we saw in CSF, it could distinguish probable possible cases as well as definite cases. Definite cases has more of this CHIT1 or chitotriosidase 1 in the CSF compared to the probable or possible cases. So again, your science, if you do something, that has to be reproduced by others. Otherwise, it doesn't give you happiness. Maybe it worked in your lab, the conditions were different in other labs, so it didn't work. Our work has been reproduced. It's just, this is a list as of mid-2019. More than 10 different groups all over the world, they have reproduced our finding. So a biomarker, unless and until it participates in the pathogenesis of the disease. So does this CHIT have any role to play in the degeneration of motor neurons. And we wanted to find out which cell in the nervous system, in the spinal cord, produces this chitotriosidase. We found that neurons not producing it, astrocytes not producing it. And in the end, we found out that microglia, which are specifically labeled with IBA1, this is antibody specific to microglia, and CHIT was labeled, the green labeling. The normal condition, there is maybe a very, very low expression, but nothing significant. But when the microglia are challenged with ALS-CSF, they start producing immense chitotriosidase. So activated microglia starts producing a huge, this is a merged image showing both IBA1 as well as chitotriosidase, abundant quantity of chitotriosidase. What does it do? The chitotriosidase is produced by microglia. What is its role? And we found out that when you add chitotriosidase to cultures, it again acts on microglia itself. It does not increase the number of astrocytes, but it resulted in microgliosis and activation of microglia. And the microglia start releasing microvesicles. These are pathogenic vesicles. And they carry all these pro-inflammatory cytokines, reactive oxygen species, and this can act on other microglia also and activate them. So here, I'm just trying to show you a movie. When CHIT1 is added to microglial culture, you see here, focus here, the microvesicles are generated. And the microvesicles are indicative of that the microglia are transformed to M1 cytotoxic type. Also, the CHIT1 does it. And does CHIT1 have any role in vivo? When we intrathecally injected the newborn rat with chitotriosidase, sometimes I say chitotriosidase, sometimes I say CHIT. And this is the normal control. And when you inject with CHIT1, the green label for microglia and the red one for astrocytes. There was a microgliosis as well as an increase in the GFAP activity. The ALS-CSF is a positive control. So these histograms show that addition of CHIT increases the microglial number more than what ALS can do. But when it comes to GFAP intensity, ALS causes more reactive astrogliosis compared to CHIT. So what happens to motor neurons when we flood the spinal cord with the CHIT through intrathecal injection? This is the cholinesterase transferase labeled motor neurons and neurofilament SMA31. And when we injected just buffer and CHIT at C500 picogram, you can see there is a substantial reduction in the CHIT labeled neurons. And this is the ALS positive control. ALS, where again it shows that there is a reduction in the chat labeled motor neurons. CHIT reduces chat. Chit-chat. So CHIT reduces chat. So it shows that it can activate microglia, make them M1 type, toxic type. It can make them to release the microvesicles, which is a bad news. And also it can directly work on motor neurons and reduce the number of cholinesterase transferase positive motor neurons. So what is our predictive model? I don't like this animation, so I just press. So CHIT1, the protein, can act on microglia through TGF-beta receptors, or toll-like receptor, or other receptors which we do not know. through SMART pathway or MAPK pathway. NF-kappa-B may also be involved in it. And in the nuclear transcription factors levels were increased. And these, because of the increase in certain transcription factors, the production of pro-inflammatory mediators is enhanced and they get released. Reactive oxygen species, INOS, IL-6, TNF-alpha, COX-2, these themselves, these can activate astrocytes, and a vicious cycle is set up. Activated microglia, activation of astrocytes, and further activation of microglia. So the production of ROS, INOS, IL-6, TNF-alpha, and COX-2 keeps on increasing because of this vicious cycle, which is set up by the activity of CHIT protein. When these toxic substances are increased enormously, what will the poor motor neuron do? It succumbs. This is our functional model. So I told you that, we wanted to do translational research, getting the inspiration from Dr. Paintal, who said that your research should not chase only the papers and citation index, but also it should do something for a human disease. It should have an application value. So I said in the beginning, translational research involves transformation of basic research into development of new drugs, new therapeutic modalities, and new biomarkers which would objectively diagnose the disease and explain its pathogenesis. I think we have succeeded in 3. So there is still a lot of work more to do. And without these people, nothing could have been achieved. I'm just a spokesperson. As some friends call me, I am a used car salesman. I can sell anything. Thank you so much.
Really, I'm grateful, sir, for your enlightenment. Thank you once again, sir.
1 minute. I just like that our Honorable Vice Chancellor must comment on your work. Over to his.
I can't hear you.
Yeah, he's listening. So I want his comment on that. He's a physician. So let him. He handles many of the neurological cases also. Let him say about your talk.
Am I audible, sir?
I think it's switch. Yeah, देखो.
Yeah.
Okay. I am basically a physician clinician. In my era, I used to handle all varieties of systemic disorders, including neurology, as a general physician. I had an opportunity to work in NIMHANS for 3 months as a postgraduate, and after getting postgraduate while working in district hospital, I was posted to NIMHANS by government of Karnataka. I saw the system, how the NIMHANS functioning is there, and it has expanded much more now. When I visit, I could not remember where the hostel I was staying. I could not locate it.
Next time when you come, we will locate it. Yeah.
Yes, sir. There are 2 aspects for me, what I learned today out of your talk and Dr. Paintal's the birth anniversary, 95th birth anniversary celebration. One thing, present students, what they feel is in whatever subject they have scored more marks, they want to go for post-graduation there with a branch which is lucrative. We could see where Professor Paintal was studying in King George Medical College, Lucknow, though he had a high meritorious the scoring, his choice was for the physiology, and he went on doing PhD, and his ultimate objective was to serve the humanity by giving new knowledge He created the new knowledge, and by that knowledge, he created so many co-mentors so that India can become rich in producing researchers of human values. Today, I am seeing from your mouth that researchers should not end up in publishing the articles alone. That publication should lead to producing new knowledge which is productive for the sufferers and bringing down the suffering of society as well as the patient sector as such. This is the value of Professor Paintal, who never bothered about his recognitions or anything at all. Though our young researcher, Dr. Sri Lakshmi, told how many medals, recognitions, Padma Shri, Padma Bhushan awards were there. And during your talk, sir, I am really impressed. I heard earlier about olfactory conduction you demonstrated in our institution in one of the conference, Physiology conference. And today I am hearing really a disease which is an enigma till today. Till now I have around 29 patients of ALS. They move up and down from Neurons to Bijapur, from Bijapur to Mumbai, from Mumbai to other neuro center. Even one of my patients went to Lucknow to get some sort of help which he can get, because it is supposed to be one of the renowned neuro center in India. You have shown us the research should ultimately end up in identifying the real pathogenesis of the disease. After identifying the pathogenesis and what pathology it produces, then whether it can be reversible or not. That is second. Once if it is reversible, what are the modalities of treatment which clinicians can use?
Yeah.
We are very much successful in the first part in demonstrating by using CSF of ALS patient, then injecting into the models and producing the model of ALS. And particularly the suffering patient, Alexander, you showed how his disease progressed, how he became
within 2 years.
Because of his strong willpower, he continued to work till the end, and his wife was more careful to give his support for his own life. And I think my young researchers have got much more motivation from you and your talk, and I want them to inculcate this habit of finding out the basic principles of disease process as such. And physiology, I hats off, because this is one of the subject which I love to my heart. And the most of the practicing physicians, if they know 3 subjects well, they can be good clinicians. One is physiology, particularly applied physiology. Second one is applied pathology. The third one is applied pharmacology. Ultimately, the first, second, and the third are the byproduct of the first. Until and unless the physiological basis is known, pathology as well as pharmacology remain a bystanders waiting for the breakthrough in advancement in physiology. We are really thankful for your valuable lucid lecture which you gave to us, sir. And I must congratulate the particularly participants who are offline. They are online, and my researchers are sitting here. We are really thankful for you for an eye-opening session which you gave to us today for our university. I thank you once again, and all the participants which have taken part in this session of observing birth anniversary of Dr. Paintal. Thank you, one and all.
Thank you. Thank you so much. And it has been a pleasure giving a talk. Actually, I prepared a talk only on Paintal before. Then, I heard that someone else is going to talk about Paintal. Then I said, I will talk about my work. And because, this is translational research is something which Paintal also wants people to be involved.
None less than you can give this talk, sir, because you are the awardee of ICMR
Paintal chair. Yes, Paintal chair.
That is one. And your work in basic neurology is worthwhile, and you have brought the translation research.
Yeah, it's it will help early diagnosis of the disease, so we can intervene better, and that's what we are also excited about.
A city like ours needs much more blessings from people like you, sir.
We are all team members. We are all equal.
Okay. Thank you very much. Thank you, sir.
Thank you for giving the opportunity.
Just 1 minute, sir. Yeah.
Thank you, sir. Now I request Honorable Vice Chancellor, sir, to present a certificate of honor to Dr. T Raiju, sir.
It will come and display it. We'll be sending you by post the hard copy.
Oh, thank you.
Thank you, sir.
It's not coming. Maybe my slides are coming. Yeah. No, I'm not seeing. Yeah.
You just, but we are hearing you. We are seeing you.
View options.
Just zoom it. Anything where the view option.
What should? Yeah, I got it. Got it. Got it. Yeah, I just got it.
Okay. Okay.
We are still learning.
Yes.
Personally, I don't like this webinar. No, you need to see your audience. You need to get the feedback cues from them.
Yes.
Then you change where your tone,
Yeah.
vary your space of lecture.
Order of the day is we have to adjust somehow.
Yes.
Till this pandemic vanishes and no more breakthrough in new disease comes like this.
Okay. So when is the end of the disease going to come? What is your opinion?
Only God only has to tell us, sir. As it stands now, probably 2021 also, it may gulp.
Mm,
A country like ours, where of course European countries are getting second wave now. India is still in first wave. And when the second wave starts, we don't know. And once the second wave starts, we heard that it is much more lethal than first one. Second infection, third
but
infection.
fortunately, the mortality rate is coming down, no? Even in US with the second wave.
Yes, sir. Yes, sir.
Yeah.
Those who have to die, they have already died, sir. Don't know.
Yeah.
Thank you, sir. Now I request Professor Kusalke Das to walk down the memory lane.
At the onset, I must thanks to Professor T Raiju, a role model for me always since long period of time. I know Professor Raju sir and also the organizing committee, the research club, Honorable Vice Chancellor, my dear colleagues and my dear students. I like to say that down the memory lane of A S Paintal, it's a bit emotion for me because his last conference he attended, that is in Bijapur, 2003, the Physiological Society of India annual meeting, along with my supervisors, my professor, B.N. Kole. But I saw him when I was the first year postgraduate student. So along with my professor, first time when I saw him, that is in Calcutta, he came to receive honorary Doctor of Science, honorary causa from University of Calcutta. And we came to know from Professor Kole that he'll be visiting his laboratory after the convocation is over. So we, the first year students of postgraduate, many other people, students and faculty, they're eagerly waiting to see Professor A S Paintal. He entered when I do not know, when we able to enter to Professor Kole's laboratory. That is one of the finest laboratory. I do not know where Professor T Raiju, who is also who is very close to Professor B.N. Kole, he knows him personally very well. Whether he has seen his electrophysiology laboratory or not, it is Paintal himself has used to tell this is the second laboratory next to DPCS Institute, where the research in receptors, Professor Kole used to work on the cardiac nociceptors and gastrointestinal receptors is done. So we went to Professor Paintal to see it. He was in a very casual dress and very smart enough, and he entered, he was asking, he was sitting with all the students surrounding, my senior, all PhD scholar, postdocs, research associates, but no faculty. Actually, he entertained mainly the PhD scholars and the researchers, young researcher. And he was going on discussing and he was asking, where is Bishu? Bishu, he used to fondly call my teacher, Professor B.N. Kole, whose name is Vishwanath Kole, as a Vishu. Where is Bishu? Where is Bishu? And he was observing everything there. This is the first meeting with that man whom we admire. And routinely we used to hear from Professor Kole when he used to deliver that class. Professor Kole, my professor, his style of teaching is always citing the reference and works of the scientists. Like that, he used to bring it from the Paintals, Devanandan, Kalia, like everything he used to tell by the name of science, Eric Nails, like that way. So whenever he comes to Paintal, he becomes emotional. So the man with whom Professor Kohli is so much of emotional, so he's standing in front of us is the first memory of mine to Professor Paintal. And he discussed about the science and all. And we are curious to know about the J receptor because we did not understand anything of the Paintal's work during that period, even though we are being taught so avidly by our teacher, Professor Kole, but still we could not able to understand the gaps of Professor Paintal's work. But beautifully, Professor Paintal has explained about the J receptor. We asked, please sir, tell us what is the J receptor and how it works. Because we could not understand it. We are reading and reading, writing in examination because we did not understand. The man in front of us, he explained, the man who discovered the J receptor, he explained to us that's the phenomena whose work is cited in the Samson Wrights and whose works is cited in the Best and Taylor, Gannon, and I'm seeing the man is explaining J receptor. He was telling that when the vessels, why it is called juxtapulmonary capillary, and why he removed the word capillary and put it, because that was the confusion in the David Whitteries. Initially, we thought that it is the pulmonary vascular receptor, which I think Dr. Silakshmi nicely pointed out that later he found it is in the atrial, right and left atrial wall, he found it, the receptor, which explained and reflexly control the blood pressure, hypotension it develops when it is filled, and the sensation goes to an afferent way to the respiratory system. This link has actually made a phenomenal changes in the various diseases as far to understand detail. So he said that when there is a exercise, he actually explained the J reflexes, which actually Deshpande and Devanandan has actually been done under his supervision in petal chest. That how it usually works, that he said that it is when there is a pressure, there is a signals goes and high pulmonary blood pressure and flows, and it causes the expansions, and interstitial fluid is full of fluids, and lungs and alveoli and as well as the capillaries in between the fluid accumulation expand and stimulate, and that's the receptor they found in here, which has been ultimately observed through the anesthetic when specific receptor stimulating drug when he used it. So he explained that this make a reflex, and this reflex normally he said that beautifully control another phenomena which he given full credit to Devanandan, Marcus Devanandan, that this had been to the muscles by which the stretch it has been changed, and this changing of the stretch in the muscle actually is an inhibitory afferent neurons, and this makes the muscle to stop its stretching, and that is the reason why lung cannot be expanded to infinity by inhalations. It stopped at that moment and control, and why exercise ultimately inhibit and make a tachypnic syndrome rather than others, and make the whole lung function modulated. We understood the J receptor from the man who discovered J receptor, which actually the Physiological Society UK has said that this is the post-Paintal era, started actually from the, though it is the single fiber technique, but J receptor make the first discovery, unique discovery in the post-independent Indian original. Even it is said that it has been next to the Ronald Ross discovery in medicine, the history in Indian next original discovery is the J receptors. So such is the man when I met him first and listened. And next meeting I had with him in his institution when I visited, with typical dress of Professor Paintal, half shorts with a white, and he was wearing a banyan, and he was very busy specs. and he discussed a few minutes with us and offered a cup of tea, and we went. So that's the painter. And later, many times we met. I met him personally. He visited here in Bijapur when I invited personally. We all in, and my supervisor took a lead role to bring him to Bijapur and grace the occasion. So, and with his wife, Professor Dr. Ashima Anand, and together we had a lot of time, emotion when he died before the NIMHANS APPI conference on 21st of December. He expired in 2004. So there are a lot of memories with him, because I can remember my professor V.N. Kole, his wife, Professor Dr. Mrs. Jyotika Kole. Immediately when I remember Dr. A S Paintal, it is emotional bondage with him and all. And later many story we heard about him, but I have seen the person sitting on the table and using the hands by which, looking at the stereodissection microscope and watching on the top where cathode ray oscilloscope is there in the old system by which he did the works. So that is Professor A S Paintal. So my greatest tribute to him, and also great regards to Dr. T Raiju, who is also a role model scientist for any Indian, one of the finest neurologist and neurophysician, neurophysiologist of India and abroad. I think this is the right occasion and the right person here as a grace the occasion and make it enriched. So my thanks to once again the research club, and specifically my thanks to my student Dr. Srilakshmi, who was my student. Now she is awarded PhD, and explains in brief about the great Professor A S Paintal story or history of discovery of science, a new generation of medical science we have imbibed. Thank you very much.
Thank you so much, sir.
I request if anyone want to speak about Dr. A S Paintal or share, anyone who are live and who are available can say 1-2 words. If anyone want to speak about Professor A S Paintal, you can unmute your mic. You can unmute your mic. Okay, thank you very much. I think we have to now hand over to Dr. Jyoti.
Now I request Dr. M.S. Biradar, sir, to say a few words.
Just to say this. Thank you.
Okay.
Thank you, sir. The essence of all beautiful art is gratitude. Now it is the time of vote of thanks. First of all, I start by giving glory to the Almighty God for making today's occasion a resounding success. I thank our special guest, Dr. T Raiju, distinguished scientist, chair of ICMR, who despite his busy schedule has found time to grace this occasion. We are grateful to our Honorable Vice Chancellor, sir, and Dean Faculty of Medicine for their continuous encouragement. I also express my heartfelt thanks to Professor Kusal K. Das, sir, and Dr. Manjunath Aithala, sir, and Dr. Sumangala Patil, ma'am, for their continuous encouragement. I thank all the participants, and last but not least, I thank IT department for their constant support. Thank you, one and all.
Thank you. Thank you very much. Thank you, sir.
Thank you, sir.
Thank you, sir. You can log off now, sir. Thank you.
Thank you very much, sir. Very good.